Experiments are a very, very common method within psychological research. So when we talk about studies like the one done by Perry and colleagues, or we even talk about Husserl, we are talking about experiments. And in this lesson, I'm going to dig into the features of experiments so that you are better able to understand how an experiment is designed, what is the purpose of an experiment, or how is it used in research. Right, so the main thing about any sort of experiment is that it's always trying to figure out whether there's a cause and effect relationship between X and Y. So does X cause a change in Y? And X and Y are called variables because an experiment is trying to understand or discover the causal relationship between two variables. Now, variables, remember, a variable is something that can vary, something that can change. The variables are either manipulated by the experimenter or measured by the experimenter, and we'll talk about that next. But for now, remember this, that any experiment is very focused on a causal relationship between two variables. And secondly, you need to remember that for any experiment, you will always have a hypothesis. Now, this hypothesis is a statement that predicts what the experimenter thinks they will find. There are three kinds of possible hypotheses that you will come across when you read research that has used the experimental method. So you will either come across a null hypothesis where the experimenter says that there will be no relationship, null meaning zero. So the experimenter does not think that there will be any kind of causal relationship between X and Y. Then second, you've got the one-tailed hypothesis, which is also called a directional hypothesis. And with a directional hypothesis, the researcher predicts not only that there will be a change, but will also predict the direction of that change, or in other words, the researcher is very clear about how X is going to affect Y. Then you've got the two-tailed hypothesis, and in the two-tailed hypothesis, which is called a non-directional hypothesis, the experimenter has not identified what the direction of the change is going to be like, which is caused by X on Y, but does specify that yes, X will cause some kind of change in Y. So any one of these three types of hypotheses are tested in an experiment, again to see whether X will cause a change in Y. Now think about an experiment that you might be able to do yourself. What is the first thing that you would do? You would first of all, of course, develop a hypothesis based on your prediction of what the relationship is between the two variables you want to measure. But you have to define those two variables first, right? And that's where operationalization is actually step one. In operationalization, researchers will define their variables clearly, the ones that are part of their experiment, or rather, let's just say X and Y for now, for simplicity's sake. They will define the variables and what the measurement of those variables looks like. So that's the first step. Now, when we're talking about variables X and Y, we're essentially talking about an independent variable, which is X, and that independent variable X is going to act on the dependent variable, which is Y. So your independent variable X has an impact on your dependent variable, which is Y, and the point of your experiment is to test whether there is indeed an impact, whether X does indeed cause a change in Y. Which brings me to this next type of variable, which is called extraneous or confounding variable. You have to design an experiment in such a way that you're absolutely certain that it's actually X which has caused a change in Y. Because if any sort of irrelevant factor gets in the way of your experiment, that irrelevant factor, let's call it CV now for confounding variable. If CV gets in the middle of this, then it might just be that the confounding variable is responsible for the impact on your dependent variable and not your independent variable, which is X. So to understand this better, let's talk about this watering can and this plant right here. Now, let's say you want to test whether mineral water will make a plant grow faster. Your hypothesis then becomes, and let's define it, right? So you say that your independent variable is mineral water and your dependent variable, you said plant growth, but how do you define that? Let's say the height of the plant is your dependent variable, and you're going to measure this, of course, by using a ruler. So you start by procuring two identical plants. One plant, let's call it plant A, is going to receive the mineral water, which is your independent variable, and plant B is going to receive normal, or let's not call it normal, let's just call it tap water. So as you proceed with your experiment, you're probably wondering, well, what do I call tap water? This is going to be called a control condition. And we'll talk about the control in a little more detail later, but for now, just remember, you've got two types of conditions for your two plants, right? So one plant is going to receive mineral water, which is your independent variable, which is part of your hypothesis that mineral water, your independent variable, is going to make your plant grow faster. The height of the plant that receives mineral water will grow faster, right? But faster than what? That's why you have to have this plant, the second plant, B, which receives tap water, your control condition. Now, both of these plants should be in a place where they get equal amounts of, oops, it's going to make a flower, equal amounts of sunlight. If both plants don't receive equal amounts of sunlight, and if, let's say, plant A receives a lot of sunlight, right? Plant A receives a lot of sunlight and it receives mineral water, but plant B receives not a lot of sunlight. Let's just color it half so you can remember. Doesn't get a lot of sunlight, but it does get the same amount of water, just gets tap water. How do you know that plant A is not growing faster because of mineral water? It's growing faster because of sunlight. So in this case, your confounding variable right here, this pesky confounding variable, is sunlight. So the sunlight here could be the cause of faster plant growth. When you compare the heights between plant A and plant B, you cannot be certain that it was the mineral water or your independent variable that actually caused the change, because it may well have been the amount of sunlight, and that is your confounding variable. And so in any kind of experimental design, the key things you got to remember are you'll always have an independent variable which should be operationalized in your hypothesis. You will also have a dependent variable also should be operationalized in your hypothesis. And then you have to design your experiment in such a way where you eliminate, prevent, control for confounding variables. So if you redid this experiment, you would again make sure that both plants received equal amounts of sunlight to prevent the fact that sunlight could very well be responsible for the height of plant increasing, right? So control for your confounding variable. Hence, plant B receiving tap water is a control condition which doesn't just provide you something to compare your mineral water plant height with, it also is something that allows you to actually determine that yes, any change in Y or the height of the plant has certainly been caused by X and not by excessive sunlight or anything else. So everything between plant A and plant B has to remain the same except for the independent variable, which will only be used for plant A. So that's how you design the most basic experiment. Now let's look at this control condition in a little more detail.
Quick pause. If you're finding value in this explanation, there's so much more to discover at Alt Academy, where we break down every A-level concept with the same clarity. Now back to the video lesson.
So the reason controls are so important in any experiment, as I just mentioned, is that it makes it possible for the researcher to ensure that any change in any change in Y, which is the dependent variable, is actually caused by a change in X, which is your independent variable. So when you're evaluating any kind of experiment, the first most important thing you need to do is to ensure that you look at the kind of experimental controls that have been used. So you would want to look at the conditions that were present in the experimental setting and the control setting, which is a group of people who do not receive the independent variable. We'll talk about that next, but for now you've got to make sure that the conditions of the experiment remain the same. For example, the light in the room, the sounds, the setting, the general sort of space in which an experiment happens must be carefully controlled. And then the second thing you need to do is to ensure that the researcher has accounted for the potential confounding or extraneous variables that might interfere in that relationship between X and Y, so that you can ensure that it is actually X changing that is causing a change in Y or your dependent variable. And lastly, now this is where control groups come in, and we're talking about them next. They essentially provide information or data that can be compared with the experimental group or the group that is actually impacted directly by the independent variable or X. And that comparison then helps the researcher to be able to draw an actual conclusion about how X has caused some sort of effect on Y or your dependent variable. So now let's talk a little bit more about this control group and what it looks like. Now the control group and the experimental group are both pulled from the same sample. As you can see, both of these groups of participants are pulled from the same sample. This is important for considerations of reliability. Remember, reliability is consistency, and it's also important for considerations of generalizability in the sense of how a study's findings can be applied across different populations. So now the experimental group is the group that will receive all conditions of the independent variable. Independent variable is applied to the experimental group. On the other hand, the control group will not receive any form of the independent variable. So both these groups, experimental and control, are drawn from the same sample, but the purpose of the control group is to provide some sort of benchmark to show that yes, it was actually the IV that was delivered to the experimental group that caused a change in whatever the dependent variable is that was being measured. In our earlier example, our plant that receives tap water is our control group, and our plant that receives mineral water is our experimental group. And so control group provides a basis for comparison with the experimental group, and it is also a way to ensure that the result of the experiment, which shows what kind of change has happened because of the independent variable, is actually valid, in that the researchers were actually measuring the effect of the IV on the dependent variable, and that no confounding variable got in the way. So control conditions exist for comparison purposes, control groups exist for comparison purposes, whereas when we talk about experimental controls, we are talking about conditions remaining the same so that extraneous variables are accounted for and eliminated or kept the same, so that your results can easily remove any effect of the extraneous or confounding variables. Now we talk about standardization, which you'll remember is very important for our idea of reliability. Now, standardization simply is that every single procedural step that is used in an experiment has been consistently applied to every participant. It remains the same for each participant. So instructions, tools, even the setting of any sort of experiment must be uniformly applied across each group of participants. For instance, in Perry's experiments with the comfortable interpersonal distance paradigm and the choosing rooms experiment, even though there was one group that received a placebo and did not receive oxytocin, and that was the control group, both groups participated. The experimental group that received the oxytocin and the control group which received a placebo participated in both of these experiments in exactly the same way with exactly the same protocols in place. In Bandura's study, which combined observations with the experiment of a model behaving in aggressive ways, the aggression demonstrated by the model remained exactly the same for every single child. There were specific steps that the model would take in the same order for every child that was exposed to the model's behavior, and the toys that were presented to the children remained consistent and uniform. The setting was exactly the same different adjoining rooms were used for each child's different trials, so on and so forth. In Hulsel et al.'s mindfulness-based stress reduction course, in their experiment, the course remained the same, and the 5 facets of mindfulness questionnaire was uniformly applied across each and every participant. Similarly, in Pizzulo et al. Study, both children and adults saw the same videos with Diego and Dora. They were shown the exact same photo array with the exact same filler task questions in the middle. So there's a high degree of standardization in the sense that the experiment's procedures are clearly laid out and then they are uniformly applied across each group of participants. This is a way of once again ensuring that the experiment can be replicated, yes, but it also ensures that the change that the experimenter is trying to identify, or rather that causal relationship that the experimenter is looking at between X and Y, is actually measured through the experiment. So putting everything together, whenever we have an experiment, first of all, you need to know any experiment will only determine cause and effect relationships which are based on this testable statement called the hypothesis. That relationship of cause and effect is expressed through the hypothesis, which may be a null hypothesis, meaning that the researcher predicts that there won't be a relationship. It could be a one-tailed or directional hypothesis where the researcher predicts what kind of change will be caused or the direction of the change, and that usually happens, by the way, when there has been previous research. Or you may have a non-directional or two-tailed hypothesis where the researcher predicts that yes, the IV will have some sort of change on the dV. So there will be some sort of effect, but the hypothesis does not state what kind of effect there will be. And by the way, in some studies in the syllabus, you may notice that there is no clear hypothesis. So you can use the term alternative hypothesis for such studies when you're talking about the aims. Now, any experiment will contain an independent variable, which is your factor x, and that independent variable or x is manipulated or influenced by the experimenter to see what kind of change or effect it has on y, which is your dependent variable. The dependent variable is dependent on the independent variable for any kind of change. Another key feature of experiments is that they will have a control group or condition, and that control group or condition will provide data for comparison with the experimental group which receives the independent variable. Controls are very important to all experiments because they help to eliminate the possibility of extraneous variables interfering with the results of the experiment in order to ensure that it is actually the IV that is causing an effect on the dV. And lastly, all experiments must have standardized instructions, settings, tools that maintain consistency for replication and retesting, and ensure that all participants, whether they are in the control group or in the experimental group, have received the exact same procedure in the exact same way. So these are the key features of experiments, and you've got to keep them in mind whether you're answering a design a study question or you're evaluating one of the experiments in the syllabus.
Hey there! If you found this A-Level Psychology lesson helpful, why don't you head on over to altacademy.org, where you can sign up for the full A-Level Psychology course. In addition to psychology, we offered 7 other A-Level subjects. Thousands of students have already taken the leap. What are you waiting for? I want to see you there.